am telling you. And these questions also you write it in your fair notebook. So everyone, I hope the students who are present just now, they all have done the 11 questions in the notebook of the chapter Cell, the fundamental unit of life. If you have done, write it in the chat box. Yes, why you write down those who have completed 11 questions. Okay, Damini, Pushkar, Archie, Romisha, Aditi Sahu has done, Ansh Chaudhary, very good, Harish Kumar has completed. And what about other students? Because I am seeing 22 here, isn't it? But only 7 students have written. What about others? See, next now, whatever questions I will give today, including those questions, you will be sending the PDF tomorrow by evening. Okay, of all the questions, answers which you have written. So you can write down quickly the questions of the membrane transport which you will be doing. Number 1, write down, define the following. Define the following. Number 1, diffusion. Number 2, osmosis. Number 3, endocytosis. Number 4, exocytosis. Noted everyone? One student can say, Romisha.
Yes, ma'am.
Romisha, repeat what I said.
Define the following: first, diffusion; second, osmosis; third, endocytosis; and fourth, exocytosis.
Okay. Now write down question number 2. Question number 2. Write the differences between end osmosis and ex osmosis. End osmosis and ex osmosis. Question number 3. What do you mean by plasmolysis? What do you mean by plasmolysis? When does it occur? When does it occur?
Can you repeat?
Ma'am, please repeat.
Okay. What do you mean by plasmolysis? Plasmolysis. Plasmolysis. When does it occur? So this, if you have read the chapter, so then you will be able to answer it. So this question you will be reading the book and answering. If you don't understand, then tell me. Now next question. What are the different types of solutions? What are the different types of solutions classified on the basis of tonicity? Classified on the basis of tonicity. Classified on the basis of tonicity.
Ma'am, repeat once again.
What are the different types or 3 different types of solutions classified on the basis of tonicity? So Safia, quickly repeat the question what I said just now. Safia left. Okay. Pavan, read the question what I have given just now.
Yes, ma'am. What are the different type of solutions classified on the basis of tonicity?
Tonicity, yeah, T-O-N-I-C-I-T-Y. SITY, tonicity. Okay. So these many questions you will be writing. Now I will start with the next cell organelle. So we started with the cell organelle. Nucleus we have completed. Now today we will take up the structure, that is electron microscopic structure of the mitochondria and the chloroplast. So now I'm starting the presentation. Can you see my
Ma'am,
screen, children? Yes,
Ma'am.
ma'am, what are the structural
yes,
components
yes.
of nucleus?
The structural components of nucleus are number one, the nuclear envelope or the nuclear membrane. Then we also call it as the karyotheca. Then second will be the nucleoplasm. Number
Ma'am,
three, chromatin network. And number
your
four,
ma'am, your voice is not
nucleolus.
clear.
Okay, my screen is visible to you.
No,
No,
ma'am.
ma'am.
No, ma'am.
No, ma'am.
No, ma'am.
My screen is also not visible.
No, ma'am.
No,
No,
ma'am.
ma'am.
Not visible.
No, ma'am.
Okay, because I'm showing you the nucleus. Okay, I'll stop sharing and once again I'll share. It's coming now.
No, ma'am.
No, ma'am.
No, ma'am.
It's
No, ma'am.
still the screen is not. Just wait, maybe little time it will take.
Ma'am, now it is visible.
Ma'am, now
Ma'am,
it's
it's
visible.
Yes, ma'am.
Yes,
Yes,
ma'am,
ma'am.
it's
Now
It's
it's,
visible now.
it's visible now. So it's taking time, that means it's slow. Okay, so now, so is it clear, children, the different structural components of the nucleus? Clear to
Can you repeat?
everyone? The new, when I say the structural components, children, I'm showing you the same thing again. The structural components are number one. Can you see my screen now?
Yes, ma'am.
Can you see this?
Yes,
Can
ma'am.
you see the slide? Nuclear
yes, ma'am.
organization.
Yes, ma'am.
So the
Ma'am.
components
Yes, ma'am.
are number one, number one nuclear envelope, number two nucleoplasm, number three chromatin network, and number four nucleolus. These are only, that is the parts of the nucleus only, we call it as the structural component of the nucleus. Did you describe the structure of the nucleus, everyone?
Yes,
Yes or
ma'am.
no?
Yes, ma'am.
Oh, okay. So now I take up the next organ quickly. So we will start with this. All we have completed the nucleus, nucleoplasm, chromatin. So did I explain about the chromosomes? I have to tell you about the chromosomes, children, today. So now try to understand. Last period I have ended up with the chromatin threads. So I told you all that in the nucleus, the network is present. You can see here. This is the structure. In this is the nuclear envelope, that is the
Ma'am, your
covering of the
voice
Ma'am,
is
your
not
voice
nucleus,
is
and
clear.
not.
Ma'am, your voice is not clear.
voice is going. Okay, okay. Wait now. Is it clear now?
No, ma'am.
Am I audible?
No, ma'am.
Not clear at all. Oh,
I'm a bit audible.
again? Is it?
Ma'am, it's
Can you
clear
hear me,
sometimes,
children?
not sometimes
अच्छा
not.
तो means disturbance is there. तो some disturbance is there, that means. And slide is clear?
Yes,
Yes,
ma'am.
My
ma'am.
Yes, ma'am.
slide is
Yes, ma'am.
visible? Okay. So try to revise the structure. In this nucleus, you find the fluid is filled, which is known as nucleoplasm. And in nucleoplasm, we find the threads are there. And these threads, they form a network, which you call it as the chromatin network. Now, when you see this chromatin network, so these threads are there. These threads are made up of the chemical substance, which is known as the DNA. What is the full form of DNA? Can anyone tell?
Deoxyribonucleic acid.
Nucleic acid.
Now, this deoxyribonucleic acid, that chemical forms those threads, and threads, they are wrapped around these globular units. Can you see this figure? My cursor is visible to you?
Yes,
Cursor
ma'am.
visible to you?
Yes, ma'am. Yes, ma'am.
It is wrapped around the globular units. These units are the histone proteins. And this way we find that the threads are forming the beaded structure. Now what happens when the cell has to divide? This is the structure which we are making, that is the internucleus. Okay, when the cell has to divide, you find that these threads, they condense. And after condensation, they form this way the rod-shaped structures, which are known as chromosomes. So chromosomes are having the
Ma'am, your voice is
chromatids.
not
If
clear.
those voice is not clear, see now some network problem is there. Try to listen, okay? Try to. So you find that this rod-like structure, this is the chromosome. And chromosome is made up of two chromatids, and they are joined together by the centromere. So you can write down the chromosome consists of the two rod-shaped structures, called chromatids, called chromatids, which are joined in the center, by centromere. 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 Noted, children?
Ma'am, can you repeat?
Okay. The chromosome is made up of the rod-shaped structure called chromatids. CHROMATIDS. Chromatids, which are joined by the centromere. 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 And then add up that they are formed by the condensation of the chromatin fibers. How the chromosomes are formed, is it clear to everyone? It is formed by the condensation of chromatin fibers during cell division. Noted everyone?
Ma'am, can you repeat
Okay.
last
It
line?
is formed. It is formed by the condensation of. It is formed by the condensation of formed by the condensation of chromatin threads. Can you see children what I am writing?
Yes,
Yes,
ma'am.
Yes, ma'am.
Condensation of chromatin threads during the cell division. During the cell division. During the cell division. Clear to everyone?
Yes,
Yes,
ma'am.
Yes, ma'am.
Okay.
yes, ma'am.
So now that was about the. Did we discuss the nucleolus, children? Nucleolus, you all have understood what is the structure of the nucleolus? Did you
Yes,
note down?
ma'am.
Yes or no?
No, ma'am.
Okay. Functions also you have noted. Nucleolus we have not done.
No, ma'am.
Ma'am.
Okay. Okay. So you can write down nucleolus. It is the site for biosynthesis of ribosomal subunits. Write down nucleolus. It is the main site for biosynthesis of ribosomal subunits. It controls cell division and replication. It controls cell division and replication. Write down, it is spherical. It is spherical.
Ma'am, can you repeat?
It is the shape you write down now. That was the shape of the nucleolus. It is spherical. It is spherical. In shape. It is spherical in shape and is made up of. So try to see from the slide, children, the last point, and is made up of an amorphous matrix. Amorphous matrix, chromatin fibers and granules. Chromatin fibers and granules. Write only that much. Now the size may vary. You can write down the cells which are actively involved in protein synthesis have large sized nucleolus. Have large sized nucleolus. Noted, children? So size will depend upon what? The size of the nucleolus will depend upon its activity. If the cell is very active in protein synthesis, it will have large size nucleolus. And if the nucleolus is not, if the cells are not active in protein synthesis, so in those cells, you'll find the nucleolus will be small. Is it very clear? Size, you understood the shape of the nucleolus and number. Remember, most of the cells will have That is, the nucleus will have only one nucleolus. But hepatocytes have two nucleoli. Not nuclei, it is nucleoli. Okay, plural form of nucleolus. Noted? So this way we have completed with the structure of the nucleus. Now I'll take up the next one, so you can write down mitochondria. Now put the subheading in your notebook, mitochondria. So you know that it is a very important cell organelle. How many students know the function of mitochondria? Can you tell? What is the function of mitochondria? Yes,
Energy
anyone.
Energy synthesis.
Correct. The production of the energy takes place in the mitochondria. So why the energy is produced in the mitochondria? What process
What?
goes on there?
Cellular respiration.
Why you are cellular respiration? And what is cellular respiration?
Production of ATP.
oxidation of
How ATP is? Anyone? How ATP
oxide,
is produced? What process going on? Correct. Again, repeat. Yeah, repeat
oxidation of
once again.
ma'am,
Oxidation of food. Oxidation of food, yes. So mitochondria is the site of cellular respiration. So note down quickly that mitochondria, it was discovered by Bollinger, and mitochondrial name was given by Benda. Okay, and it is seen in all eukaryotic cells except old RBCs. Except old RBCs.
breaking in between.
Yeah.
Ma'am, your
Old
voice is
RBCs
breaking.
do not have. I'm not able to get your voice again.
Ma'am, your voice is not clear.
My voice is not clear. The same way your side also the voice sometimes is disturbed. All the time the voice is like that, or you find sometimes it is disturbed.
Ma'am, sometimes.
Yes, ma'am.
Sometimes it is getting clear and sometimes it is disturbing. Okay. But from my end, everything is okay. Some network issues must be there. Okay. Now try to see the structure of this mitochondria, children. Can you see the screen, all of you?
Yes, ma'am.
Okay. So the mitochondria, they are spherical, oval or discoidal. Write down the subheading shape and note it that they are spherical, oval or discoidal. Now you write down the subheading number. How many mitochondria will be present in a cell? Cytoplasm. Okay, so you find that the number varies and depends on the metabolic stage of the cell. If the cell is very much metabolically active, if it is a very active cell, so the mitochondria will be larger in number. And if the cell is less active, you will find that there will be less number of the mitochondria, because mitochondria is producing the ATP you said. So if the cell is functional, so then it will need more energy. That's why you will find that more number of the mitochondria will be present in such cell. Okay, now try to see the structure. So put down the subheading structure. Each mitochondria you find it is bounded by two unit membranes. Mitochondria is bounded by two unit membranes. We call as outer membrane and inner membrane. And between the outer membrane and inner membrane, try to see the structure. We find the space is there. That space is known as intermembrane space. What is the space called? Can anyone repeat?
Intermembranes.
Intermembrane space.
Intermembrane space. Now, you find outer membrane is smooth. but the inner membrane is projected inwards to form these finger-like projections, which are known as cristae. What are these finger-like projections called?
Christe.
Cristae. You want me to write out the spelling? It is cristae. Cristae. Cristae are what? They are the finger-like projections. Okay, finger-like projections of the inner membrane. Is it very clear to all? So now try to see here. You find in this diagram the outer membrane is smooth, but the inner membrane is projected inwards and it is forming these finger-like projections, and you call these finger-like projections cristae. And inside the inner membrane, now one more thing, remember these cristae, they are having the, now I'll show you the other slides, it will be clear to you now. This F0 subunit and F1 subunit. Could you see this? On this cristae, we find these small structures are there which are known as F1 particles or oxysomes. The cristae are having oxysomes or F1 particles, and they have this structure, that is, it consists of the stalk-like structure, that is F0, and then this is the F1 part, the globular part of the head is there. So the cristae have the F1 particles. You write down that way. Now inside the mitochondria, can you see here the matrix is filled. This matrix is containing the genetic material, that is DNA and ribosomes. So write it quickly, everyone. The matrix contains DNA, ribosomes, and enzymes. So these are the enzymes which will be used up during the cellular respiration process, that is during the oxidation of the food. So write down that enzymes needed for the oxidation of food. So what are the
Please
structural
repeat.
components of the mitochondria? I'm repeating the whole thing now. The structure, the mitochondria is bounded by two unit membrane called outer membrane, which is smooth. Outer membrane, which is smooth. Then the other one is known as the inner membrane, which is folded or projected inwards to form the finger-like projections called cristae. Look up the spelling if you don't know. Cristae. This increases the surface area for respiration. Inside the inner membrane, so next component, you write down the matrix. So inside the inner membrane, the matrix is filled, and this matrix is the homogeneous fluid containing containing the DNA, ribosomes, and enzymes needed for cellular respiration. Understood, everyone?
Yes, ma'am. Ma'am, can you repeat
So,
the last line?
the details. Last one. The matrix contains, it is a homogeneous fluid and it contains DNA, ribosomes, and enzymes. enzymes needed for cellular respiration. Okay, so you can note down the number. Those children who want to write down that the mitochondria contain more than 70 enzymes and the coenzymes. Okay, and they are all needed. They are responsible for converting the energy stored in food to ATP. Okay, so mitochondria is known as the powerhouse of the cell because there is continuous production of ATP. And remember, we call mitochondria autonomous organelle. Why you call it autonomous organelle, children? Because
Because
it
it has its
is
own DNA.
having its own DNA and it has the ribosomes that can do its own protein synthesis. It can do the protein synthesis. So that is why you are calling it autonomous organelle. So shall I move to the next organelle now? It's very clear, I hope, mitochondria.
Ma'am, can you explain again?
I'm saying that it is known as autonomous organelle. Autonomous मतलब जो खुद कर सकता है, isn't it? So here autonomous meaning that it can synthesize the proteins itself. Okay, it has whatever proteins are needed by the mitochondria, it will prepare itself. Okay, why it is able to prepare the proteins? Because it is having the DNA, it is having the ribosome. Very clear now?
Yes,
Ma'am, what
ma'am.
was the
Yes, yes.
ma'am matrix?
Matrix is the fluid. Okay, matrix is the fluid which is filled inside the mitochondria. So that fluid is containing the enzymes and that fluid is only having the DNA. It is having the ribosome. Just now try to understand जैसे cell के अंदर. Cell is bounded by the plasma membrane and then inside the cell you find the fluid is filled which you call it as the cytoplasm. Same way the mitochondria is bounded by the two unit membrane जिसको अपन outer membrane और inner membrane बोल रहे हैं और उन दोनों के बीच में space भी है जिसको अपन inter membrane space बोल रहे हैं. ओके और इनर मेम्ब्रेन तो फिर फोल्ड हो रहा है फर्दर एंड फोल्ड होके फिर क्या बनाता है वो फिंगर लेग प्रोजेक्शंस एंड दैट इज नोन एज द क्रिस्टल. फिर उसके अंदर फ्लूइड है, उस फ्लूइड को हम बोल रहे हैं मैट्रिक्स. इज इट क्लियर टू एवरीवन नाउ?
Yes, ma'am.
एंड देन आई टोल्ड यू अबाउट द कंपोजिशन ऑफ द मैट्रिक्स. तो मैट्रिक्स में क्या क्या प्रेजेंट है? तो मैट्रिक्स में डीएनए है, मैट्रिक्स में राइबोसोम्स है, मैट्रिक्स में और क्या प्रेजेंट है? कैरियल
Enzymes.
Enzyme.
Enzymes.
enzymes. And more than 70 enzymes and coenzymes are present. And they all are needed during the process of cellular respiration, that is oxidation of food. Now we start with the next cell organelle. So write down everyone, plastids. Plastids. So plastids are the organelles which are present only in the plant cell, children. Okay, and they are of two types. What are those two types? The chromoplast and leucoplast. Two types of the plastids are you can write down the plastids. So what are the types of the plastids? Number one, leucoplast. Leucoplast, now remember that they do not have the colored pigments. Do not have colored pigments. They do not have colored pigments. So they help in storage. What is the function of these leucoplasts? They store the food material. Function of the leucoplast is to store the food material. Some leucoplasts will store the fats, some will store the starch, some will store the protein. So this way the different types of the leucoplasts are present in the plant cells. The plants are doing the photosynthesis and they prepare the food. So that food is stored in the different leucoplasts. The second type, we call it as chromoplast. Chromoplast, and chrome word means color. So these are the colored plastics. They have different colored pigments are there. They have different colored pigments. They have colored pigments instead of plastics. You write down colored pigments are there. Is it very clear? So, out of these colored pigments containing plastics, which you call as chromoplast, one only we will be studying which contains the green color pigment, and that is known as chloroplast. Which pigment do the chloroplasts have?
Chlorophyll.
Green.
pigment.
Green pigment, chlorophyll.
And what is the pigment name?
Chlorophyll.
Chlorophyll.
Chlorophyll. So this everyone knows that the chloroplast is having the pigment chlorophyll. Everyone noted this much? So shall we move to the structure of the chloroplast? So we will study what now? The structure of
Chloroplast.
chloroplast. Okay. So now I'll take up little today and then we'll So now try to see this structure. This is the chloroplast. It is also bounded by two unit membrane. So first structural component, number one, you write down membrane or the covering, membranous covering. So in the membranous covering, then you write down that it consists of two unit membrane, the outer membrane and inner membrane. Can you see the figure, children? The outer membrane and the inner membrane. And they are not sticking to each other to two membranes. So you find the space is there, and that we call it as intermembrane space. And inside the inner membrane, we find again the fluid is filled, and that fluid is known as stroma. What is the fluid known as? Stroma.
Stroma.
So first component will be the membranous covering. Second structural component is stroma. So what is stroma?
Ma'am, the fluid present.
So the fluid which is filled inside the chloroplast, it is known as stroma. And this stroma, write down what it contains. It contains starch granules, osmophilic droplets, and enzymes. And it also has the DNA, children. You know that two organelles are there which are autonomous organelles. Okay, that is your mitochondria and chloroplast. So it has the stroma, also has the DNA and the ribosomes. DNA and ribosomes. So all have understood what is the composition of the stroma? What is the composition of the stroma? Can you repeat?
Ma'am, will you please repeat?
I'm saying the stroma consists of the starch granules, osmophilic droplets, DNA, ribosomes, and enzymes. DNA, ribosomes, and enzymes. Needed for what? These enzymes will be needed for which process?
Photosynthesis.
So write down. Very good. So write down enzymes needed for photosynthesis. Needed for photosynthesis. And then write down in the next line that stroma, or it is the site for the dark reaction of photosynthesis. It is the site for the dark reaction of photosynthesis. Clear to you, everyone?
Yes, ma'am.
Okay. Now the third structural component, that is lamellae. You write down third structural component, lamellae. The spelling is given here in the slide. You can write down lamellae. 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 Now there are two types of lamellae. The third structural component is lamellae, and these lamellae you find they are of two types. One is grana lamellae. Grana lamellae. Now what is grana lamellae? All have noted grana lamellae?
Yes, ma'am.
Okay. Now you try to see in this diagram, these, can you see these sac-like structures? In this diagram, these sac-like structures.
Yes, ma'am.
These sac-like structures, which is labeled here as thylakoids. Could you see in the diagram? So these sac-like structures are the thylakoids, and these thylakoids you find they are placed one above the other. Just as we are placing the coins one above the other, same way the thylakoids, which are the sac-like structures, they are placed one above the other. And when they are placed above the other, they form one unit, और वो sticks of coins जैसे appear होते हैं, and you call that structure only as granum or grana lamellae. What is it known as?
Grana laminae.
Grana lamellae.
So you write down grana, they are staked lamellar structures. Okay, and what is it consisting of? Thylakoids. So I'm dictating, you write down grana lamellae. It consists of the sac-like structures, and take up the spellings from the screen. Consists of the sac-like structures called thylakoids, which are placed one above the other. Thylakoids which are placed one above the other. Forming granum. Forming granum. Forming granum. Done, everyone? And these thylakoids contain the chlorophyll pigments. Thylakoids contain chlorophyll pigments, pigment molecules. And write down what is the function of it. So, it is the site of light reaction. It is the site for the light reaction of photosynthesis. So we'll end up the class, children. The next part I'll tell you in the next period. I'm stopping the sharing now. Yes?
Can you repeat the last line?
It is the site of light reaction of photosynthesis. So I'll send the question. I'm ending up now.
Ma'am, what were lamellae?
Lamellae, the whole structure we are calling it as the lamellae. So grana lamellae or the granum is the same thing. ओके, एंड देन द स्ट्रोमा लेमिले इज देयर. जो वो कनेक्ट होते ही ऐसे ग्रानम जो एक के ऊपर एक जो थाइलाकॉइड अटैच होते हैं, फिर वो कनेक्ट होते हैं ट्यूब लाइक स्ट्रक्चर से. So that we call it as the stroma lamellae. So I'll further discuss next period. Okay, so I'm stopping the recording now.